Hi, my name is Katherine Thompson-Peer. I'm an assistant professor in the Department of Developmental and Cell Biology. I'm going to give you a little flavor of our work today. My lab is currently accepting rotation students, and hopefully you'll want to come join us. So we're interested in this question about how neurons, nerve cells, respond to injury. And so to give you a little bit of background, there is a long and extensive history of studying how nerve cells respond to injury to their axons. So go back to intro bio. The structure of a nerve cell is there's a cell body in the middle, and this is a nerve cell that I'm going to show you here, so I'll just point to it. There's this cell body in the middle. There's a highly branched dendrite arbor. So this is the receptive antenna for that nerve cell, where these neurons receive information. So if they're receiving information from the environment, if they're sensory neurons, that's where they detect temperature or pressure or light. If they are neurons in the brain, that's where they're receiving information from other neurons in the circuit. So there's this highly branched dendrite arbor, cell body in the middle, and the axon is a long cable that allows the neurons to send information to other neurons or target cells that can be really far away. So it can be a nerve cell in your spinal cord that extends an axon all the way down to your toe. And those then signal to the next neuron in the circuit. So maybe they form a synapse onto a muscle cell to induce that muscle cell to contract, or maybe they form synapses onto other neurons to inform how cognition happens in the brain. So taking this kind of basic idea of the neuron's architecture, there are many years of study about how neurons regenerate axons. And lots of people who are working on it in a variety of different systems, in mice and rats and humans and flies and everything. But there has been remarkably little work on the question of how neurons regenerate dendrites after injury. And both of these types of architectures are equally important for how a neuron functions. It both has to receive information and send it along. So a neuron that's been injured needs to be able to recover both of these functions in order to be able to work properly. And so my lab is interested in this question of how do neurons regenerate dendrites after injury? And we've been using the peripheral nervous system of the fruit fly Drosophila, in part because it's super straightforward to find an individual neuron and follow how it responds to injury. So I'm going to take you through just kind of a basic experiment that we do. This is an uninjured control neuron, and so we look at these neurons, and this is 24 hours after it would have been injured if it was an experimental neuron, and 72 hours after it would have, but this is just the uncut control neuron. And you can see that between these time points, that neuron grows, it becomes a little bit more complex, it adds a lot of terminal branches, but the overall architecture of that neuron doesn't change. What we do is we use a 2-photon laser to selectively and specifically cut off all those dendrite branches. So it's on a confocal microscope, and we focus that laser on those branch points and increase the power and use that to specifically injure dendrites and ask how these neurons recover. And so this is a neuron 24 hours after we cut off all of the dendrite branches. There's the cell body in the middle and the axon going down, and we call this balding when we cut off all of the dendrite branches. And you can see that that territory that neuron was responsible for covering is empty. These particular neurons are polymodal sensory neurons, so they respond to a variety of different environmental cues. They respond to light and temperature and touch. And you can see that here they don't have any branches to extend out to their segment of the animal's body where they're supposed to detect light and temperature and touch. But if we come back to these same neurons a couple of days later, if we come back 72 hours after injury, you can see that the neurons regenerate relatively robustly. They elaborate these very complex dendrite arbors that have a whole lot of branches. And this had already also been seen previously. But what happens if we look a little bit more closely is that you can appreciate that these regenerated and the uninjured neurons are not exactly identical. So they, in some ways, the neurons regenerate very robustly. They regrow these branches that look like dendrites, that function like dendrites. If you count them, they are the same number of branches as an uninjured neuron. They've got the right proteins that are being segregated out into the dendrites, separated away from the axon. But you can appreciate also that this arbor is a whole lot smaller than the uninjured neuron. The branch arbor is much more compact. The branches cross over one another. They're much thinner. And so what we want to do is kind of address this question in two ways. What is it that allows the neurons to regenerate as well as they do? Because regeneration is certainly impressive, but what is it also that holds these neurons back from regenerating perfectly? Why can't they perfectly recreate what an uninjured neuron looks like, both in terms of cell intrinsic and tissue environmental factors that are preventing them from perfectly recreating what an uninjured neuron would look like and perfectly restoring that function? So if we look at another different class of neurons, you can kind of see this even more strikingly. These particular neurons, there's the cell body in the middle and the axon going down, and you can appreciate that they have these long primary dendrites and then the short actin-rich terminal spikes. And with these particular neurons, when we cut off the dendrite branches, here's a neuron that's regenerated after injury, you can see that it regenerates, but it really only regenerates these terminal actin-rich spikes. So one of the things that we're interested in investigating is what is it that keeps the neuron from being able to regenerate these long primary branches so that it can only regenerate the terminal actin-rich spikes, but not recreate kind of the primary, secondary, terminal sequence of things that would allow it to extend branches across long distances. Another avenue of exploration is trying to look at how these neurons interact with the other cell types that they are in contact with. So here on the left are outlining the sheet of epidermal cells that the neurons are growing out their dendrites on top of. So here we just got a junctional protein that is outlining all of them. And the neurons grow on top of that. And so it's known that the epidermal cells communicate to the neuron, signal to the neuron to decrease or dampen dendrite outgrowth and development in later animals. So one of the things that we're interested in studying is, can we improve regeneration by getting rid of some of this inhibitory signaling that might be coming from the adjacent epidermal cells? So far, I've been showing you experiments in Drosophila in the fruit fly larva, which is often where we start because those experiments are more straightforward. That's this animal on the left that's expressing GFP only in these cell types that we care about. But we've also got expertise doing these experiments in the adult fruit fly, which allows us to address the questions from slightly a different angle. How does aging relate to dendrite regeneration? How does dendrites regenerate in a stable animal where they're not normally changing, where they're established? The lab also has other projects going on in terms of what is the role of chromatin remodeling in dendrite regeneration? What is the role of known regeneration pathways specifically in dendrite regeneration? What is the role of ER calcium homeostasis in dendrite regeneration? You're probably sensing the theme. So with that, I'd like to say thank you for your attention, and please send me an email if you have any questions or want to come talk. I'd like to say thank you, of course, to the members of my lab. We are small but growing. I currently have 2 graduate students in the lab, a technician, a co-mentored graduate student, a couple of undergrads, and we're looking for more people. These are our funding sources, past collaborators, and thank you so much. Have a nice day.